Right. Welcome to the VacCAP webinar series. My name is Amaya Tucha, and I'm a member of the VacCAP team, and I will be today's webinar moderator. Today, we will be discussing BerryPortraits, phenotyping of ripening traits with automated imaging tools and softwares in Cranberry using YOLO V8 with Dr. Janine Loarca. Dr. Janine Loarca is a recently hired USDA Agricultural Research Service research geneticist working on blueberries and strawberries, serving the Pacific West area, and bringing with her over 10 years of experience in special crop breeding and genetic research. Her program prioritizes traits that are critical to blueberry stakeholders, such as machine harvestability, environmental resilience, and fruit quality. We're really excited to have Janine tell us about this BerryPortrait new tool. And so thank you very much for Janine for doing this. I just want to remind everybody attending that to please stay muted and with your camera off during the webinar so we don't distract the speaker. If you have any questions, feel free to just put them in the Q&A or in the chat box, and we will be discussing those and reviewing those at the end of Janine's presentation. So with that, I'll turn it over to Janine to start her webinar.
Thanks, Amaya. That was a very generous introduction. It's great to be in a room with so many people that I had the opportunity to work with on Cranberry during my postdoctoral work, and with whom I get to continue to work with in blueberry and small fruits moving forward. And that's kind of the spirit of this software paper that we just published last week is how do we bring together a resource that can service a lot of small fruits that have similar traits that are important to their marketability. So I'll start with some acknowledgements. I was part of the Salapa lab during my postdoctoral fellowship and the Atucha lab. I had the opportunity to collaborate with the P.E. Marucci Center for Blueberry and Cranberry Research and Extension in New Jersey. We have some USDA folks, some Rutger folks, and then Breeding Insight was really wonderful for driving this work forward and co-creating the software with us. So I'm presenting on this paper. This paper was, a lot of the authors on this paper are on multiple of the institutions that you see here. And this collaboration is something that was really critical to the success of this project. And it's something that I see as critical moving forward to have an interdisciplinary group of plant scientists, breeders, horticulturalists, software engineers, thinking through and conversing about what traits matter, how do we phenotype them accurately, how do we reduce the cost of phenotyping. So when I originally came into my postdoctoral work, I was interested in characterizing genetic variation for fruit quality traits in a cranberry diversity panel. After looking around for a couple of weeks, it was clear that the goal was going to be to develop a high throughput method to characterize the genetic variation for fruit quality traits, because the method that we had, which I'll talk about later, wasn't performing as expected. So let me first just talk about the name so you get a sense for what we were trying to do and what we're doing with the software. So why portrait? A portrait is a painting or a photograph, a visual representation of a person, something that's trying to capture their likeness, their personality, and mood. Except in this case, our subject isn't a person, it's a berry, and we're trying to characterize their likeness, their essence for all these various traits. And as you can see here, there's variation for a lot of traits that you can see visually in Cranberry, and we want to be able to characterize and quantify those traits. So this is an acronym that just came about while sitting around on a Sunday morning. So the first 4 letters of this acronym, BerryPortraits, is phenotyping of ripening traits. So when I'm referring to ripening traits, I'm thinking about the traits that growers use to visually assess the readiness of their crop for harvest. In Cranberry, that tends to be color and size, and uniformity of color and uniformity of size. Because I felt like being a little bit extra, I was like, oh, wait, we can add some more letters to this acronym. So with rapid automated imaging tools, so what that means is that we're using a camera, a light box, and a computer to record the images and store them. And then the S, these are the kinds of images that were generated by the Salapa Lab and by Jeff Nehart's group. And if I'm forgetting anybody, I'm sorry. And then finally, the S is for the software. And this was the core of the collaboration with Breeding Insight, was developing this software that can read the images and extract phenotypic information from them, and then give us an output file that we can then statistically analyze to make decisions about culling or selecting. So this is open source code. It's available for anyone to use. You can go to the GitHub website. The website's down here. You can find the code over here in green. And two people that were with us to develop this were Chris Strock initially, and then it was bittersweet to lose Chris because then we got to work with Tier, and Tier has really been instrumental in building this. And we've had conversations with him every couple of weeks for the last 2 years to figure out, okay, how do we build this software in a way that's user friendly and accessible to more plant scientists and usable for cranberry. So that was and so the traits that we're measuring are here. They're RGB, your LAB traits, object area, perimeter, width, height, surface area, volume, and then we can also extract from these measurements uniformity metrics. And that's what I want to talk about right now, because we're used to saying getting like maybe mean trait values or variances, but we're focusing now on developing it's not really developing, but just using a statistic to measure the uniformity of the traits that also matter to growers. So, for example, if we wanted to measure the uniformity of the color, this program allows us to do that. So the way that I think about, so if you imagine that these four blocks represent four unique cultivars, we can see that they're different, but how do we characterize the difference that we see? So on the horizontal axis, I like to think about that as being berries that have multiple colors within a berry. And then as you go to the right, you have berries that are more uniformly colored within a berry. It's a trait that I called intra-berry color uniformity. And then if we focus on the other axis going up, down, if you compare all the berries to one another within the photo, do they look more similar to each other or do they look dissimilar to each other? That's a trait I called interberry color uniformity. And so this is the thought is then that if we can quantify inter and intraberry color uniformity, we can make good selections that serve our stakeholders who want uniformly colored, uniformly ripening berries. And we can do the same thing for size. There is an interest, of course, in size uniformity and in high fruit weight because it correlates with improved processing efficiency. And cranberries, 99% of the product is, maybe it's 95% of the product is ends up being processed. So we can use these selection indices to then combine how red do we want the berry to be. Let's select for the highest uniformity of color, similarity within a group, high fruit weight, similarity of fruit weight, and proceed with selections that way. I also want to say that we're not new kids on the block doing something that nobody has ever done before. These are some other phenotyping softwares that have been developed over the years for small fruit crops. I added hops to this because this was a recent publication, Hopbox, that also uses Python. So what I'm saying is if you're trying to find something where you want to do high throughput phenotyping for your crop, maybe explore these options, see if they'll work for you. Not every program measures all traits, but I think there are some advantages to our program. So our program was built actually on top of or inspired by the one on the second row here, Gina, which was developed in the Salapa Lab in 2016 by Luis Diaz Garcia. And it was a great product. It didn't really, it had some wins, but it didn't really take off. It had really good, like the internals of the program were really great. And this is from his 2016 paper where he's showing that you can segment cranberry, you can measure cranberry, but you can also do cherry and potato and pumpkin seed. Like this program had really good generalizability. And it was used by a number of publications to measure in blueberry, also in cranberry and some other miscellaneous crops. And unfortunately, it didn't get widely used in the last few years. And so part of our goal was to take the internals of GINA and put it in a more user-friendly format. And that's something that Breeding Insight was really instrumental in helping us to do. And we also wanted to make sure that our program was consistent with the quality of data that GINA was outputting. And that's largely what we did in our paper. We wanted to see for the traits that BerryPortraits measures and the traits that GINA measures, are we getting the same values? And largely we are, which are clear because of the very high correlations that you see here. So those are for color metrics, and these are for some of the shape and size metrics. Very high correlations. And then we wanted to also see if the regression relationships were the same among traits between the software. So having similar correlations among traits was a good indication to us that these programs are picking up the same signal. It also allowed us to hone in on areas where they're not quite picking up the same signal and going, what's going on there? Or, oh, that's fine, that makes sense because we made an improvement to the calculation. So we had some ideas about why GINA didn't take off, and we've identified a few barriers to access. For one, GINA is built in MATLAB, which is proprietary software. It's not mainstream for plant science researchers. We don't work within that to do our data analysis in grad school largely. MATLAB is a steep learning curve for most plant science researchers, and the program, GINA, is no longer actively maintained. So even if it produced a great product and good data, it was difficult for folks to use this product. So as we were working with Breeding Insight, we were thinking, how do we overcome these barriers? How do we build bridges to access? And these were some of our solutions that I think we implemented. So the first is, instead of using proprietary software, we're using open access and open source software. So BerryPortraits is in Python. And the source, as I mentioned, the source code is available on GitHub. Anybody could go there right now and download it. To overcome the steep learning curve, we put it in Python, which is more widely known in the data science world and perhaps more familiar to plant science researchers. And Breeding Insight is constantly maintaining and adding new traits with our input. The data integrates with Fieldbook and BreedBase. And because we have this active community of researchers, we can respond to feedback quickly and improve the software over time based on needs that are identified. So I want to put out there to folks here that being that this is open access, we have this license, a CC-BY license. This license gives you the freedom to use this software, to share it, remix it, adapt it, distribute it, reproduce it. You can even do this commercially. There's no restrictions in that kind of way. So take this, have fun, adapt it to your program if you think you can use it in your crop, in cherry or in anything. If you have colleagues that you think might benefit from this, feel free to share it with them. We just ask a couple of things. Okay, 3 things actually. We just ask you to give us credit if you publish, so make sure to cite us. Provide a link to the Creative Commons license to the CC-BY 4.0 license. And if you made any changes, indicate what the changes were that were made. So to give it a little bit more specificity, make sure to give credit to the original paper, the source code, so the GitHub page for Breeding Insight, and then here's the link for the Creative Commons license. My question to you now, and this is meant to be a discussion, how would you and your team imagine using BerryPortraits in your program? What additional functionality in BerryPortraits would be useful for your program? And what would make you or your team more likely to use BerryPortraits? This feedback is really helpful for us because sometimes it's just a minor modification that results in this program being able to be adapted to like citrus, for example. And so if it's something minor like that, it's perhaps worth some investment on our part to make the tweaks. If it's a huge thing, you might be on your own, but there are a lot of resources. It's a really nice learning community for Python. And yeah, I just encourage folks to be creative with this and play with it and don't be afraid to learn and don't be afraid to fail. And um, yeah, it's just an invitation to be part of the growth of this program. So this is my contact information. These are some crops that I imagine could be phenotyped with BerryPortraits. And I just want to talk to folks in the group now and get your feedback about what you would like to see happen with this next. So thank you.
Thanks, Janine. So given that Janine wants to have a conversation, I think that, we're a small enough group that we could have people unmute themselves and ask questions about the BerryPortrait software, or also just provide some feedback on where you think you could use this tool. You can also just, if you don't want to unmute yourself and ask the question, I'm happy to read it if you type it in the chat.
Hello. Can you hear me? I'm sorry, I'm on my phone.
You're heard.
My name's Alex. I'm the ag scientist for Ocean Spray in Washington. It's actually really interesting. I was shared this imaging system randomly, but I have been like prototyping in my free time a very similar, just the photo box, not the software. And been taking pictures of fruit using different little 3D printed trays. I haven't read the paper yet. I was wondering if you could speak a little bit to the support for building the physical platform, the hardware, getting your camera system set up, and getting the trays so the fruit's in exactly the right place and things like that.
Yeah. So there's very detailed information about that in the paper. What I can tell you is that the hardware is stuff that you might already have laying around your office or your lab. So we needed a poster board, we needed a light box, we needed a SLR camera, we needed a color swatch, or yeah, I'll just say color swatch for now, and then some scale markers. And then we wanted to make sure that the fruits weren't touching, and amongst our labs, we all had different ways of making that not happen. So I think Jeff Nahart used like a poster board that was divoted slightly and let the berries fall into them. We created this, it was just like a piece of a board, and we bore a 5x5 holes into them, laid them out, and then put the fruit in those holes and then pulled it back. And yeah, and then it was essentially good to go after that. I think it's a pretty low input system. It's controlled lighting, so being aware of what your light source is and keeping track of that will help maybe to color correct, especially if you're using a different kind of light bulb across imaging sets. And I want to, because we are talking about hardware and photography, I do want to invite Edwin Rydell, who is here and who is in Breeding Insight and was part of this work, to comment further if there's anything more that you think could be said about that.
Oh, hey, Janine. Wasn't expecting to be on video. Okay, looks like I'm presentable here. Hi, everyone. I'm Edwin Rydell. I'm the phenomics coordinator with Breeding Insight. And I think Janine covered most of the basics of an imaging setup. And I guess I would say we'd be happy to help you out with that if you have any further questions offline. But like Janine said, having a good quality light source and having a single light source is pretty important. Taking images, even simple things like white balancing your camera before you start taking images, taking raw images if you have an SLR versus going straight to a JPEG format, which is a compression that is lossy, and the information that you lose, you might actually want. Yeah, and then there's lots of tools online for calibrating, like for your camera setup, your sensor and your lens. You can calculate how far you need to be from the subject to get everything in focus, to get everything in the frame. Those tools are widely available. So yeah, lots of little things, but once you get going, it's pretty easy. And another thing, Jenny, that you didn't mention was I think an improvement is we had some difficulty with the reflections on the fruit. So there's some opportunities for improvement there as well.
We have a couple of questions in the chat, and maybe Janine and Edwin, you both can help answer these. The first one is, would the background need to be the exact same for us to be able to use this software? And if not, can there be ways to remove the background variation?
Lushan, thanks for that question. That's something that we thought about a lot because we had images from Jeff Nyhart's program and from the Salapa Lab program, and the Salapa Lab program, and even in different years, and folks would change the color of the background. I would say when the model is being trained, which is something that Tier did, he used a variety of images, and just having a diverse set of images helped actually. And so I don't think it would be too much of an issue if the background isn't the same for every photo. I would caution though to try and select a background color that contrasts with the color of whatever your crop is. So for example, we found that with our more yellow cranberries, being on a white background was problematic. The software couldn't always find where the edge of where the cranberry ended and the white background began. So we would get just these weird kind of blob shapes around certain berries. So yeah, I would say before you launch into like a full scale, I'm going to do this on 300 genotypes, try some different backgrounds and do some tests on some and see which backgrounds are giving you the best segmentation results.
We have another question here in the chat. It says, is the camera distance and field of view fixed? And would this be an issue for photographing larger fruits such as grapefruit?
So in our case, yes, the camera distance is fixed. And I think you might need a larger imaging box to the needs of your crop. You might need a larger box, or Edwin, maybe you want to comment on like the size of the size of the lens, or maybe how tall the box needs to be. Do you have any other ideas, Edwin, how to photograph large fruit?
Yeah, with a large fruit, you probably need a little more distance so that you're it's called working distance to get the whole fruit into focus. With small fruit, it's pretty easy. If you focus on one part of the fruit, the whole thing's probably going to be in focus. But if it's bigger, you might need to get a little bit farther away, use a larger f-stop, for example. So yeah, it really just depends. I'll put one of the calculators, the depth of field calculators, in the chat, and people can check those out.
Great. Do we have more questions? And again, feel free to unmute yourself and ask the question if you don't want to type it in the chat.
I'll go. This is Jim. Janine, can you use the software to look at images from outside to get an idea of ripening time and things like that from field images?
We haven't tested it, but I would be worried about the various kinds of light conditions that those images were taken under. So BerryPortraits has been tested under uniform light conditions, but cloud cover or time of year or shade from a neighboring plant can affect how the fruit is segmented or perceived at all by the software. I think if something is going to be developed for an outdoor program, it needs to be tested in that environment. So
Gotcha.
That's very similar question in the chat, asking the same thing, if this could potentially be used in the field, but it just doesn't seem to be the case with this version of the software.
There are programs available that have done field-based imaging, and if you go to the paper, I think it's in the supplementary tables and figures. Thanks, Edwin. But there are some examples that might suit your purposes.
Right.
And if that's what would be applicable in a field setting in the future with a makeshift light box. For low-growing fruit crops, I've seen apps able to distinguish color about that. So is it like a cart that's kind of enclosed, but like it rolls over your crop? Or Jade, would you mind saying more about that?
No, I'm just wondering if it's possible. I haven't seen anything like that. I'm just wondering if you think it would be possible.
I think we don't know until you try it. And I know we have a project. Well, I shouldn't say we anymore because I'm not at UW-Madison, but I know there's a project at UW-Madison to develop a cranberry cart that you can roll over, and cranberry's low growing too, obviously. So you can roll it over the plots, and it'll. They're trying to find a way to do imaging outdoors because it is a pain to do all the harvest and to store the fruit and lay them out in a certain way. It'd be really nice if we could get the same quality data, but in a more efficient way.
Yeah, I agree. That would be great. I guess time will tell. Thank you.
What crop do you work on?
This would be for cranberry. So I'm the agricultural scientist for Ocean Spray here in BC, Canada.
Cool.
Yeah.
Do we have any more questions for Janine?
Sorry, just one last thing for Jade. Jade, if you want to put like a Google alert for like Salapa, if any publications come out that are doing field-based phenotyping, you would get an alert as soon as that publication came out because he's the one that's doing that work. Although Jim Polishok is also doing that work, so you might want to also, although with Jim, you can say more. I can't. I'm not sure all the details you're doing, but you're doing multispectral, yeah?
It's in the plans. Juan's group, we're working with Juan's group and Matt Dingman at U-Madison, and development is going to be as part of the VacCAP 2 grant that we're all working on. So hopefully it's coming.
Good to know. Thanks, James.
Any more questions? I did put a link in the chat for the recording of this webinar. So if you want to go back and watch it again and listen with more time to some of the answer to the questions, you can do that. It's going to be posted in our YouTube channel for the VacCAP project.
Thanks, Amaya. I'm also putting my email in the chat. So if you are thinking about this later and a question comes to mind, just send it to me and we can have a conversation. Or if you want to follow up on something that you've asked, I'm always happy to talk about this kind of stuff.
So Janine, you're now working on blueberries, correct?
Yeah, and strawberry.
So, okay, I didn't know strawberries too. So are you going to try to use the software package for blueberries and strawberries?
Yeah, definitely. One of the things that's special about the Pacific Northwest strawberry is that it has red colored. And so it's something that would be really nice to start collecting phenotypic data on and, have more of a quantifiable way of selecting redder and redder strawberries. It's also nice data if we ever decide to do like a release, to have that table with all those kinds of values for like a hort science paper. Edwin, do I want to mention the web app or is it not available? Oh, that was a direct message. Edwin, I don't know I don't think a web app is available yet, but I know that there are plans in 2025 to develop more of a graphical user interface. And if folks use this and see stuff that they're like, could you measure this also? And if it's like low-hanging fruit, sorry for the pun. Let's talk about it. But yeah, the web app, go for it, Edwin.
Oh, we've royal we here, but BI has been working on making a web GUI interface for running the BerryPortraits. So should be available soon.
Yeah, so trying to make it even more user friendly. This is version 1, right? So like we're interested in feedback and we're interested in actually one of the questions you're going to get a survey after this, and one of the questions is, are you interested in testing the usability of this software? Drop your email and we'll start to include you when we meet up to discuss that, because ultimately we usable for as many people and as many crops as we can accurately phenotype. Yeah, thank you, Michael.
Yeah, I'm having a computer glitch right now, so it may not go out immediately, but it'll be out today sometime.
Can I ask folks what crops are represented here? And feel free to drop it in the chat. I see a strawberry, I think, in somebody's photo. Or is it just a generic rosaceae?
Cranberries, blueberry, kiwi berry.
Ooh.
Interesting. Also have
Oh my gosh,
raspberry.
nice. Cool. It's really neat to see some blueberry colleagues here who I haven't had a chance to meet yet. So one of my plans is to test this on blueberry sometime in my first year in this position. I started, I don't know, 9 weeks ago or something. So diving right in. But if you're interested, if you want to take some photos with this kind of setup, and you can send them to me and they can be part of our training set, that could be a way to, to get the ball rolling on this so we can all be using it.
And Janine did put her email address in the chat. So if I'm gonna put it again right now, but there she is if you want to contact her for following up on the software. Well, it doesn't seem like we have any more questions.
For the blueberry, I have a question.
You have a question? Maya. Well, please go ahead.
For the blueberry folks, what traits would you imagine measuring with this software?
Hey, Janine, I think that we had some conversation about that. The size will tell you, for blueberry, the size will tell you the weight, because they are relatively the same, they are highly correlated. other than the size, then the color in blueberry, yeah, maybe the color, but then I don't know. Size, appearance used to detect quality, yeah, shrivel, yeah. So probably the post-harvest, somebody's suggesting the shrivel, and maybe it will be a way to quantify on a more objective scale the shrivel. You will need to have the fruit probably for a week or at least a couple of weeks to start to see shrivel.
You know, this feedback is really useful because as I'm, you know, as this summer, as I'm collecting and putting aside fruit to photograph, to, you know, to get a training set, if I see fruit out there that have these traits, you know, I won't just pick like the beautiful ready to eat fruit. I'll pick, you know, some of these ones as well, some with defects that, yeah.
Yeah, right. So then some disease, fruit disease, then maybe, yeah.
Disease would be so interesting because, you know, well, okay, Jim's trained in pathology, but to somebody untrained in pathology like me, a lot of diseases look the same. But I bet differences in, you know, if you look really closely, this virus in cranberry has a very different expression than the other virus in cranberry. So yeah, how cool would it be to be able to train a model to identify what pathogen you have in your field? I mean, this is something that's maybe more of like a grower application for an app like this. Yeah, there's a lot of potential applications with this. Obviously, I'm in breeding. I'm a breeder, so this is what I'm thinking about it. But there are potential applications in food science and horticultural science and plant pathology.
Yeah, Hiduk pointed also stem scar size. You know, sometime we collected in VacCAP we collected with the caneberry, but maybe using an image you are more accurate.
Yeah, we use it in my lab. Well, not this version, but we use the Gina version. And one of the cool things that feature had, and I know that BerryPortrait also has, is the actual shape of the fruit, because in cranberries you have different shapes, and that was important. And when you do some like plant growth regulator studies, that can affect the shape of the fruit, and that I remember using it, and that was very helpful, because if not, you have to do everything manually, and that is crazy. So that's another trait, just fruit shape, that can be measured with this.
Hi, Janine. Can I ask another question?
Oh yeah, hi.
Hi, thanks for the talk. It was great. I was wondering if you could speak a little bit about the computational power required to run these types of models, as well as to maybe store image data, if that was a limitation.
Yeah. That's a Tier question, so I'm going to punt it to Tier, who's not here. So I don't have those numbers right now, but based on, you know, whatever system Tier was running BerryPortraits on to get the data that I then analyzed, he was able to run an image in 5 seconds.
Okay.
But I'm not sure I'm not sure what his, you know, what resources he was working with.
Okay. And how many photos did it take to train the model, did you say?
I didn't say. I think it was just over 500 between our three sets. And they all had different backgrounds. They were in different light boxes, different genotypes.
Gotcha. Thank you.
Yeah. So going back to the question that I think one of the first questions that was asked about the background variation, I was thinking to myself as well, like, do we need to get this background color down to like the hex code for it to be like super repeatable? And the answer is no. Like, working with a variety of images just it improves the model, and it makes it more flexible for users who are like, how can I recycle this gray piece of cardboard that I have sitting around in my lab? Can I use it for this? Yes, use it. Susan, can it be used to analyze leaves? I don't know. I would love for you to try it though. I know Gina was used to, looking at leaf area. Actually, Amaya's postdoc is using it for that reason.
Yeah,
right?
we're testing it to look at leaf size and leaf area, but yeah, we don't have yet a final decision whether it works or not. We are still collecting the data and then comparing it with other systems, like when you use like a regular scanner and you just use ImageJ to do the same thing.
I know the previous software that we built this off of, Gina, was used to study leaf size in Kura clover, which is a crop I didn't hear about until I read that paper and still don't really know what it is. But if our software is, if the guts of our software is equivalent to Gina, then I think there's a good chance that it could work. I would encourage you to try it and let me know how it goes for you. Would be great to get a measure of nitrogen chlorophyll content. Again, if folks think of stuff later, my email has been in the chat. I'll drop it again. I'm always happy to talk about this. We're always looking for feedback or new ways that this could be used or, subtly tweaked or optimized for your purposes. It is a USDA tool, so while the software is open source, big development projects are sort of reserved for USDA scientists. But I think a lot of what we have is generalizable enough that it could work for your crop with very subtle tweaks that we could accommodate right out the gate. But give it a try. Give it a try.
Well, it doesn't seem like we have more questions from the audience. Janine, I don't know if you have more questions.
No, I'm done.
All right. Well, we just really want to thank Janine for sharing this new tool and for taking the time to answer all the questions. If you have more questions about the BerryPortrait, just please contact Janine. And we thank you all for attending the webinar. As I said, the webinar, it's being recorded and will be posted on our YouTube channel. Please help us with providing some feedback when you received the email with the server. That really help us make these webinars more informative and better for our audience. So with that, thank you very much, Janine, and thank you everybody that attended today's webinar.